Thank you, Reia, for your speech. Nous allons maintenant échanger avec les panélistes. Est-ce que vous avez des questions?
I don't know whether they're going to speak French or English to go from one to the other. Do you have any questions? Somebody's. Yes, you can ask your questions in French. Everything is translated. Yes, I have a question. How come when the coral are in the nurseries, they have more diseases than in their natural spaces in coral reefs? How come? I think maybe there's a lack of genetic diversity. I'm not sure. I'm not sure of the answer.
The nursery areas, at least for in Guadeloupe, actually has less disease than in the transplant sites. So it's actually the reverse. And I think a lot of that comes from the fact that the nursery areas in Guadeloupe, they have different mechanisms. They have trees which are suspended nubbins, that have suspended nubbins. They also have the bioballs and wire meshes that is separated from the natural substratum. And they have tables with little cookies where you would have different species growing. So because we have some separation from, no? They will be translated shortly. Because you have some of the separation from the natural substratum, it is twofold, I think, in my belief, is there are very few vectors for disease. There are very few fish currently at any of the sites. So fish is one of the potential major vectors for disease. You also have less chance of the fireworms and the mollusks, the snails, to traverse the sandy areas to get to the transplant sites. So they actually have to make a big effort. They may smell the corals, the tissue, but it's harder for them to survive the trip to those sites. So actually the nursery areas have less disease than the natural environment.
Merci Reia pour ta présentation qui était très complète sur les.
Thank you for your exhaustive presentation of the methodologies and everything that has to do with restoration. I've been asked to ask challenging questions. I'll try to be challenging. It has to do with opening the discussion on the strategy for coral restoration. We've seen that the environment is essential, water quality, fish presence, it's things that you cannot master. But is the strategy is to develop restoration in endemic clean areas, marine protected areas, to increase restoration possibilities? Or do we have to get interested in degraded sites? Because generally for coral reef we could, but there's not much success possibilities. So what would be the good strategy for a good success? Where should we have them?
I love this question. Corals are actually very resilient in time. What we've noted, for example, in Tobago, in further south, we have a lot of the same problems of eutrophic waters, very nutrient-rich waters, heavy sedimentation, low salinity, because we are heavily impacted by the Orinoco flow. But we also have corals that survive this. It means that we may not have as great a diversity, And it may look very disastrous for a little while, where lots of corals may be dying, but the organisms that are left are those that have those genetic oopses that I was talking about, and are able to withstand many of those stressors, right? So to answer the question, for here, you can't wait to get the perfect environment to do the transplantations. Corals grow too slowly. Our fastest growing corals are the acroporids. They grow up to about 10 centimeters on average a year. The majority of corals that we have other reef building corals, the orbicellas, for example, the star corals and the brain corals, the Pseudodiplura strigosas, etc. These grow millimeters a year. If we were going to wait until we had the perfect environment and to resolve things that we know is not going to ever be resolved, we're not going to stop shipping, we're not going to stop development, we are going to have things spilling over into our water systems, we'll be waiting forever. We just need to keep trying until we, I'm going to say this in a bad way, until we weed out the weaker corals, until we get the really resilient ones, and then those ones that we will to continue to have growing in our environment to improve what's happening there. That answers it. Okay.
Thank you for your presentation. It's very, very good. Moi, j'avais une question toujours pour challenger.
I have a challenging question. You haven't spoken about the endotherms. It's very important for some coral. The symbiosis with the zoophylaceae. Could we introduce it in the complexity to improve the system?
So in terms of zooxanthellae, which is a symbiotic algae that lives in the tissues of corals, these zooxanthellae have a symbiotic relationship. They actually, for many of the species we have in the Caribbean, provides a large percentage of the nutrients that corals utilize for growth. So corals get their nutrients, their energy from two sources. They feed themselves, of course, but the majority of which lies from both the zooxanthellae as well as some microbes that exist on the mucus layers of the corals. Up until recently, it has always been suggested that the clade, the type, the variation of zooxanthellae was really important for the survivalship of corals, particularly as it relates to bleaching. Clade D, there's clades like F+. Clade D has been the focal point for the Caribbean for being able to withstand best high temperatures. There's some recent research coming out, particularly from Saudi Arabia, where it's not so much the zooxanthellae. While yes, the clade D can withstand higher temperatures, it seems that given the right conditions, the right stress responses, corals have the ability to produce a particular protein that is a heat shock protein that can protect whichever zooxanthellae that they have. They just need the right sequence of stress. So what we had started to uncover in Saudi Arabia and in the Red Sea, where you have major shifts in salinity, where you go from perhaps 15, 16, 17 ppt to 40-something ppt. So that's huge shifts. And similar major changes in temperature. And it goes back and forth, and then you would start seeing this heat shock protein being expressed by the coral. So it's not something that we automatically, the coral's automatic, we are in the coral, that switches on. But it's something that I personally have an interest in, to see if we can actually find a mechanism to switch on that gene to produce that protein to help protect the corals. But that's just one stressor. There are so many others.
Oui, alors en fait, c'est pas une question, c'est un...
Well, it's not a question, it's a testimony I have. In 2016, the National Park worked with the University of the Antilles and set up an experiment for the restoration of Acropamata coral, the hybrid that proliferate. So we had bits and pieces in the Grand Cul-de-Sac Marin, bits and pieces that we took to in marine protected area on to harvesting, or rather planting high sea and others, and after 100 days the 3 species were high in recovery. 5% for the first one, second for Trifora, and the rest for the Acropora calmata. And when we transplanted these 3 bits, well, species bits and so on, and when we used substratum, they could not resist, invaded by algae. But a few Metis area Acropora species developed quite well. We monitor them, their necrosis rate and growth, and they develop quite well. And yet it's just close. So it means that the quality of the environment was maybe better at that time. Maybe the water quality was better. So do we have to intervene? Because if conditions are good, the corals recover quite well.
I like this one too. One aspect I didn't talk about was orientation. The transplantation of the species that you're interested in transplanting, because it's not always Acropora, it's Orbicella, Pseudodiploris strugos. Those are the common ones that are used for transplantation. Orientation is important. Certain species like to be upright when they're transplanted because of the positioning of the polyps themselves to capture light. The form, so many species can grow in different forms. Acropora palmata, for example, is known to be spread out more like a little plate, kind of like an antler, which is like an elkhorn antler, which is where it gets its common name from. They like lots of light, but they also like a lot of energy. So you would find Acropora species typically in very high energy sites. They like that. They would break, which is normal, but it's one of their normal mechanisms for colonization. So choosing the right site, choosing the right orientation for transplantation is also important for these. Some of the corals that prefer these plate structures tends to be done in lower light. They tend to spread out to maximize. It's almost like a canopy of a tree, right? They want to maximize. The leaves will bend based on the light. In terms of corals, to bend a little bit might take them 10 years, you know. So they've got to choose that form for growth and be consistent with what environmental conditions they have. So transplanting a coral and transplanting it in a good orientation based on the growth form that you're using, and they are adaptable as well. But if you have more of a plate-like coral, you want to orient it. It's not just as simple as I take a coral and just slap it very crudely, attach it on the substratum with the cement mix or what have you. A little care sometimes can go a long way in their survivalship by having just the right angle to maximize the majority of light. as well as to minimize the amount of sedimentation that settles onto the polyps. So you would have polyp structures that are relatively low in rugosity, so they are, it almost feels like a Porites, for example, that has very low polyp height, versus a Cropera that has a very high, relatively speaking, polyp where they're growing up. Sediment can get trapped in and amongst the polyp structures, and that can introduce not just diseases, but it could also introduce anoxic or areas of low oxygen on the tissue structures that's going to kill off and create more stress. So orientation of the placement of the polyp, the transplants are important as well.
On est très organisé. Bon, on parlait d'espèces coralliennes, donc ça m'a fait penser à une autre.
Well, I was thinking that coral is very well organized, and yes, it reminds me of the kind of coral. Very often we choose large leaves and branches, and it works well. It's quite good to have a good figure on growth. But when you work on restoration, it's not just the coral community you want to restore, but a whole ecosystem. It's a reef. It's a coral reef. And in a coral reef, it's not just large leaves. You have massive, you have long-standing and others. So you have considerable effect on wide branches species, but maybe there's a lack or a failure in restoration because it's not genetic diversity, but forms, shapes of coral should be diversified for restoration.
Oui. For a successful reef system, what you would like to encourage is high rugosity. Rugosity, you could think about like a skyline, and the taller you have tall buildings and short buildings, the more intricacy in terms of the structure, the more rugosity that you have, the more microhabitats that's created for other organisms in your reef structure. And I somewhat mentioned it in the talk, where you want to encourage habitats for other species. You want to encourage functionality on your reef system. You want to ensure that your reef system has structures, surfaces, habitats, nursery areas for example, reef fish. I mentioned there were very few reef fish in the areas that I dove last year when I was here. And that's disturbing from a perspective of having herbivores, for example, on our reef system, especially in nutrient-rich waters, which is what we're dealing with here, is really, really critically important, because these are the lawnmowers of the ocean. Having sea urchins, having herbivores like the parrotfish, etc., and as well as a host of other species that will graze not just on the edges of the corals, but also physically remove the turf algae and the fleshy algae that's on the reef system. That is what helps give corals the edge. So just transplanting flat corals or even just branching corals, it's a start, it's a process that you learn, and you learn from mistakes. But the aim long term is that you transplant not just branching corals, but more of the plate corals, the boulder corals, but you also transplant it in such a way that you create that artificial skyline, so to speak, under the water. By doing this, even though you have no fish now, without any other intervention, eventually they will come back. Reef systems are quite resilient in that way. They will be attracted to these sorts of habitats, and by extension, having the coral reef systems being rehabilitated, and by extension, the seagrasses and the mangroves, you have this tri-ecosystem areas where nursery habitats and areas are being promoted. Fish will come back. Other organisms will come back because corals and the algae and even the smaller fish are attractive areas for food. And so what you want is to build that functionality over time. It's not going to happen overnight. But this is why microfragmentation has taken place and has become the next hot ticket, so to speak, in transplantation. Because with microfragmentation, you can get a lot of clones very quickly and recreate huge boulders or any other structure because they're going to reconnect. Corals, it's like chemical warfare that goes on between species, even individuals of the same species. If you don't have the similar genetic code, then potentially they're going to fight for space. If you have a similar enough genetic code, it doesn't have to be necessarily identical, but if by all means on purpose, by having a clone, it recognizes self, and so it will fuse. And therefore you can have very quickly lots of these microfragments that have been placed in whatever structure that you want, so that you could create these artificial structures, these three-dimensional structures, and have these corals that fuse that will basically cover those structures and simulate. So you don't have to wait hundreds of years to get to these very large corals. You can either artificially create the substratum, or you can use the dead coral surfaces that are existing and transplant on those, because we know corals used to like it there at one point, and then just recreate and recover, shall we say, the reef structures. But yes, it's really important. It's the first step, I think, towards getting and building that ecosystem resilience. So going from a speciation resilience into an ecosystem resilience is very important, yes. Long answer for quick.
Donc du coup, pour rebondir un peu sur tout ce qui a été dit finalement.
Okay, to sum up everything that has been said, environment is important, conditions are important. If you try to reduce pollutants, well, no, let's not be pessimistic, but probably we will never manage to remove all pollutants because they're there to stay. But one issue with all the diseases is that coral do not have time to adapt. Changes are so brutal, they don't have time to adapt. So if you have nurseries in healthy, semi-healthy, more or less polluted areas, maybe would enable us to select corals that would be more easily adaptable, and those in healthy areas would be moved and then create a mix of coral so that some species endangered could have a better lack to survive, a better possibility.
Yes, so by having this intermediate area that will be more easily to substitute corals into a more polluted area makes sense, because basically, like anything else, you are trying to condition the corals in a slow manner to where they're going to end up. So two things is looking at the sources and the sinks, which corals naturally have the capacity and the ability to survive, but also allow the corals the time to naturally adjust, because they are relatively resilient. For a very simple organism, they have a very, very complex genetic structure, and a large part of that has to do with their ability to produce mucus as a layer of protection. They can encourage certain bacteria to grow on their surfaces that will give them the sorts of nutrients that's going to be required to function, to grow, to repair, to build their immunity. So yes, giving them an opportunity to adapt is critical, which is why nursery areas, I think, are important. And having nursery areas, in the case of corals, in different environments is important. So bringing corals up even from deeper depths, going from shallower to for near shore to further offshore. These are all variations that we can do. It's a trial by error, which is why we only really take a nubbin as opposed to the entire coral, because we want to ensure that the stocks remain and that they heal as well from it. And we don't normally just keep breaking nubbins from the parent colony. That's why in the nursery areas you can create nubbins from the nubbins that you had from before. And that way you can build your reef, by picking and choosing which nubbins you're putting where, which genetic mixes that you're putting, and also having that variation, because Acropora, they generally, specifically for Acropora, they don't travel very far. The distance of their larval flow is relatively small compared to many of the other corals. So the genetic mix, especially for an island like Guadeloupe, similarly to Tobago, is not expected to be very great. So there's only so much we could do from a genetic resilience perspective. But what we do have, we can actually create mixes, and like I said, just create the best circumstances, the best environment to give them a fighting chance for when we do deploy them at their different sites.
It's time for Q&A. Yes. It's time for Q&A. We're going to open the floor to every one of you here, and of course online as well. So if you have questions, si vous avez des questions, please introduce yourself, speak slowly, and of course deliver the question. So si vous avez des questions, levez votre main, je vais venir vous donner le micro. Parlez doucement, s'il vous plaît, pour nos amis interprètes. Et puis ensuite, évidemment, on vous passera la parole. N'oubliez pas de vous présenter quand vous présentez votre question. Lou, je suis à votre merci. C'est à vous de me dire où aller.
Est-ce qu'il y a des questions dans la salle? Oui.
Thank you very much. That was very interesting, fascinating. I mean, it was very an interesting panel. My question was about your last point about the selection of the most resilient genetic mix. Mm-hmm. Do you think this is a solution for which you will have enough time, because you have the time for research to see what species will be more resilient, and are there also some negative collateral impacts of a selection of genetic mixes, because then you may not develop other mixes which would be resilient to other diseases that you were not researching for? So I was just thinking about. I mean, do you think this is really a future for the restoration of coral?
It's a current. It's what's happening now. There's a lot of work being done on the genetics of corals. A lot of research is currently focused on Acropora because they are the faster growing corals. In some places, like in the Cayman Islands, it can grow even as far as 40 centimeters a year. It's an anomaly based on our research. But the reason for choosing Acropora is not only that they were hammered very badly in the 80s, 82, 83, where they were decimated by white band disease and white plague, but they are also, because they're the faster growing corals, you can actually do a lot more research with them in a shorter space of time. So in the Caribbean, and as well as in Australia and some parts of Australia, they actually focus specifically on acroporate research. Now, if I kind of take that one step back, in the geologic history of the Caribbean, acroporids were the dominant corals. Currently, we have some of the bolder corals, the star corals, and the brain corals, which have superseded acroporids because of the loss that had occurred in the early 80s. And so the bolder corals, and these are like the orbicellas, so like the star corals that you would find, and the brain corals. These are much slower growing corals, but they tend to be a little bit hardier compared to the branching corals, and in this case, the acroporids. So natural selection is what we're seeing happening with the boulder and the brain corals. But we're spending a lot of time and effort trying to understand not just the genetic code, but the genotypes for the Caribbean, trying to see, especially for islands that are relatively close to one another, if there's a good gene flow, if there's similar genotypes. So if we can establish what, let's say, of the 6 odd genotypes that cervicornis has here, 6? 8 of cervicornis. Let's say there's one predominant one, genotype 3, if I'm not mistaken. Let's say that disappeared. Then we can look for other islands that have similar genotypes that we can more easily introduce from nearby islands, because the chances of that gene flow that it would have come from somewhere or has the potential to come from there is greatly increased. Of course, you're looking at it from an island perspective, but when we're looking at transplantation, when with natural gene flow, we need to look beyond Guadeloupe. We need to see where the sources and sinks are outside of Guadeloupe as well for something like this to create that. But it's very slow going because the corals do grow slowly, but they have one of the substratum techniques, which is using an electric field. This is something that has promoted growth of corals to kind of speed up the process, because it takes a lot of energy to precipitate the skeletal structures. There's a lot, when it comes to the physiology of the animal, the tissue of corals are less than 2 millimeters. It's very little to a coral. The majority of it comes from its skeletal structures. So a lot of energy gets put into deposition of that skeletal structure. In times of high stress, they want to deposit as fast as possible, which means that they're sloppy. So it's just like building a house and a foundation. If you take your time and you place every block carefully and you mortar everything nicely, it's a nice, firm, thick, rigid structure. But if you want to do it fast, you're just throwing the blocks down there. It's, there's not enough mortar. There are weak spots that happens, and you have gaps that occur. So while you might have extension of the skeletal structures, it's much more fragile. So by having there's a point to this. By having an artificial field, an electric field that aids the corals for precipitating the calcium carbonate, the energy can now be diverted elsewhere for growth and immunity and just repair. And there's some strategies as well if we wanted to help corals. It's kind of going a little beyond your question. For diseases, etc., there's some work going on in Florida where if they create a trench, where if they see a disease, for example, they create a physical scar on the corals, something that we're told, don't touch the corals, don't do a lot of things. But basically what they do is that they create an artificial injury onto the coral. And if the injury is bad enough, what the coral does is it abandons that site. So it retracts the tissue from that injury. So it says, okay, I'm going to lose that part of my tissue structure. I'm going to conserve my ability of growth. So I will pull back where my tissue is going to grow. And then it's, like you cut off your limb to be able to to escape a trap type of a thing. That's the sort of things that we're finding. So from a nutrient perspective, from an energetic perspective, the corals can maneuver their nutrients and their energetics where it needs to go. And so by allowing and providing the corals an environment that helps them do this is a good thing. And in an artificial setting, you could provide better food resources, better lighting conditions, etc., good healthy bacterial mixtures for the growth of those corals.
Thank you, Reia, for this explicit answer. Il y avait une deuxième question du monsieur avec une chemise grise.
Oui, monsieur. Alors, nouvelle demande. Please stand up. Levez-vous quand vous posez votre question. Pour les caméras, s'il vous plaît. En français ou en anglais.
Oui, bonjour, Dr. Guppy.
Good morning. My name is Thierry Petit-Legrand. I'd like to know if in coral studies, because I know what you showed is on reproduction and living communities. Are there any research on communication? Because we know that in ecosystem, in forestry in particular, we have current research to know that trees and plants have specific features. So in corals, do we have this? Is there exchanges that are not just chemical that would enable, as I said to Madame Mage, that the planting of Acropora palmata in a place is dying? And if there's a natural species that is developing, could there be something, it's not just scientific, but maybe we could see whether there's communication.
So in a nutshell, because there were some mic issues, basically, do corals communicate together like trees with mycelium? That's kind of the beginning of the question.
Yes, except it's more warfare. If it's not the right genetics, they literally use their tentacles that has little stinging cells. And I mean, if you speed up slow motion, it's like this, and they will grow until they just can't touch one another. But I like the question because one of the things for settlement of larval forms, for example, is light. And corals fluoresce. And many organisms on the reef fluoresce as well. And there's some research that we're actually trying to do now to see if we can get the right light, fluorescent wavelength, to see if we could encourage settlement. Okay. Do you want to tell before I go on? Yes?
Les polypes sont assez proches et du coup ils se frôlent les uns les autres, donc il peut y avoir une communication comme ça.
Sorry, time out. We're in a time out.
Okay. I will translate the questions then. For each question, I will translate. Je vais traduire les questions quand les questions seront posées. Très bien. Alors, y a-t-il une autre question peut-être? But don't worry about time. We have 10 minutes left.
Okay.
Ah oui, là ça va être compliqué. Basically, what I understood is that they basically communicate with each other, or they. I need to translate in French. Je dois traduire en français. Excusez-moi. Oui, c'est ce que j'ai compris. Donc, c'est qu'ils communiquent entre eux par un jeu de touche-touche avec des fibres. C'est bien ça, Lou?
Oui, les tentacules, on va dire.
Les tentacules. Merci. Moi, je fais de la géopolitique, donc j'essaye. Vous connaissez mieux les mots que moi. Et fondamentalement, ça permet effectivement de faciliter la transplantation, si j'ai bien compris, d'un territoire à l'autre. Mais peut-être ai-je mal compris, Lou.
Si, c'est ça. Et qu'il y a il n'y a pas encore beaucoup de connaissances sur ces aspects de communication, on va dire, entre coraux, mais qu'il y aurait un effet sur l'installation, en tout cas sur du long terme, des espèces entre elles ou de colonies entre elles sans...
Yes. So I'm not sure what she said, but I'll continue the story. So communication, a lot of communication happens from a larval form. And a lot of that communication is based on chemical cues as well as visual cues. So corals as a larval form, they and many organisms that have a benthic or that attach on the benthos of any system, they will come out of the water column and bounce around. And is there hard substrate? Yes. It's like, okay, I'll keep looking. Is the substrate suitable for me? Are there other corals? Are there other carbonate structures? Yes, okay, I'll keep looking. And then they're looking at the fluorescent cues as well. So there's some research that my lab is actually starting now to see if we can have artificial structures with particular fluorescence that will encourage larval settlement of corals. Once they are established and they are growing into adults, the majority of the communication is based from chemistry and from coral to coral or from coral to other organisms. It's chemical warfare. Beyond that, within the coral head, where you have multiple animals that are living as a community, there's communication. They do communicate. They can redirect resources where necessary for improving growth and protection against disease and those sorts of things.
Donc, en fait, c'est surtout au stade larvaire qu'il va y avoir des... Est-ce qu'il y avait d'autres questions? Il y avait une question de Monsieur Diaz, juste derrière.
Oui, j'arrive tout de suite.
Bonjour à toutes et à tous. Donc Nicolas Diaz, direction de la croissance bleue de la région Guadeloupe. Dr Guppy, merci beaucoup pour cette présentation, ce panorama complet des techniques de restauration.
Thank you, Dr. Guppy, for this panorama of restoration. What appears is that it is still experimental and innovation and discovery. Small scale, really. But what we know is that the impact on natural system at a wider scale show that there's accelerated decline of these natural ecosystems covering hundreds of square kilometers. So my question is, will we be on time to repair while it is still time? And shall we meet the timing and under what condition?
Is there still hope to repair before it's too late?
Well, I can't speak for the future. I could speak for what's happened in the past. And there's been environmental changes happening, maybe not as fast as they're happening now, but like I said, corals are pretty resilient. They've existed for hundreds of years, thousands of years, and they've gone through global warmings and global coolings. Not as many species may survive for the future, but what does survive, I like to think and to pray something will survive. The diversity may be different. We're already seeing that. We're living that right now. The diversity may be different. But I think the hardier ones will survive. Which is also why I think it's important to build these biobanks, these seed banks, if you will, just like we do for food storage. We have a potential to actually now invest in that for our natural environment, for corals and other organisms that we find that are going to be. I mean, you've got to use the technology that we've got available, and that's already available to us, that we can actually store cryogenically the larval forms and/or the eggs and the sperm of particular species of corals. So in the future, we can perhaps transplant from that perspective. But from a natural perspective, something is going to survive. I like to hope it's just not going to have as big a diversity.
Est-ce qu'il y a une autre question dans la salle?
Oui. Ah ben, monsieur, puisque vous êtes là.
Bonjour, monsieur Fazian de l'association Casa Recycle. Je représente une association qui, depuis à peu près 2016, a expérimenté des récifs artificiels, c'est-à-dire des éco-mouillages.
I don't know if he's going to translate, but he's talking about eco-mouillage, as they call it, meaning a ship fixed moorings at sea, ecological moorings. If you have ecological moorings in a lagoon, right in the middle, with Gautier Islands, with a coral reef suffering, does that make sense? Does the rest of the Caribbean speak about this, where you have about 40 ecological moorings, where you're going to have corals and larvae? We've been working on this in Saint Francois for the past years in Les Saintes, 14 meters depth. I take advantage of having Madame de la Casa and the port, so we started in 2017. I'm not here to complain, but we've been working for the past 7 years. We have various diving students who would like to be part and parcel of your research. What I'm interested in is all these scientists to have a concrete answer. Is it logical to have eco-moorings when you have seagrass beds that are damaged? Does it make sense to have coral reefs? Does it make sense? Yeah,
That's a long one.
It's a long one. I think if I understood correctly, ultimately the basic question is, does it make sense for...
Is it worth
it?
Eco-wetting, is it worth it?
Yeah.
And especially in areas where you've got seagrass meadows.
Well, for corals, you mean?
Yeah.
Or just anything?
Yeah, for corals, I imagine. Pour les coraux. Yeah, for corals, yeah. And for the whole ecosystem at large.
Well, there's transference. You have what we call metapopulations. Metapopulations, you could think of this as even meta-reefs as well, if you like, where you would have small systems that are spaced, that will allow movement of organisms as a whole, not just corals, just movement of organisms to go from one habitat to the next. In a positive way, this will allow reseeding and the introduction of safe places for smaller organisms, the babies, the bubbas, to get from one location to the next, to make it out into the further reef system, given the challenges that they face. There is interconnectivity between corals, seagrasses, and mangroves that's been well established, especially when it comes to commercialization, commercial fishing, fisheries, and these sorts of things. But from a habitat perspective, you do have certain species of coral, certain species of fish, shellfish, mollusks like the queen conch, which I know you have here, that they can use some of these habitats to expand their range, where they would have beforehand not been able to really do that, given all of the challenges. So I'm always of the opinion for myself to create, because we've taken away so much from our natural environments, that if we provide these artificial structures, in time, they will be able to, we may not get back exactly what we would have lost, but we would get back to another level of stability and recovery that maybe from a functional perspective be similar to what we had before. From a speciation perspective, it may be different, but from a functional perspective, they may encourage those habitats. And once they are in an environment that is not in the way of development, because let's be real as well, people need to live, we need to develop, but we also need to ensure that we do it in a sustainable manner. So having small dome structures that are intricate enough with variations to their structure will allow a multitude of organisms to come back. And once the fish start coming, I think fish are really, really important to the story because they're the ones that, for lack of a better phrase, to make it very simple, are the ones that manage the area. They maintain the structures. They're the biggest bosses on the reef system when it comes to that. So if we create habitats for the fish in particular, we will be going a long way to allow the slower growing organisms a chance for survival. It just takes time and patience. For ecosystem rehabilitation and specifically for coral rehabilitation, you have to look in decades, not in months or in a couple of years. You have to look in a decadal perspective.
Une dernière question.
Maïté Najan du Parc national de la Guadeloupe. Vous avez beaucoup parlé de l'environnement et de la...
You spoke a lot about environment and the quality of environment. In Guadeloupe, water is a main challenge. Today, as managers, we're not trying to do transplantation because for us it didn't work. So we're trying to protect what remains, hoping that one day water quality will be best. But you said that you also had in Trinidad and Tobago water nitrates issues. Managers were they more interested in water quality or restoration, or it was a mix? Because for me, I feel that in Guadeloupe, if you don't manage water quality first, you're not going to restore coral reefs health.
Yes, of course, because thank you for your question. Yes, we want to be able to, of course, improve water quality. In the case for Tobago, it is not solely anthropogenic. It's not solely based from an island source. This is something that naturally happens around Tobago from the Orinoco flow, where you would have however many millions of tons of fresh water filled with nutrients, with high in sedimentation, that comes over Trinidad and Tobago and sits over our waters for periods of time before it traverses the rest of the Caribbean. So our corals are naturally stressed from those conditions, in addition to what runoff and nutrients that we have and our ports and our shipping industry and the water quality that you would get from that perspective. So to say, if I understood correctly, that transplant, saving the corals is not really should be the priority per se. I don't really agree with that because the need for the corals to be present, yes, to try and improve water quality is a big issue, yes, I agree. And it's not the end all and be all for the corals. So the corals can survive lower quality water. The diversity is just not going to be that great. The growth is not going to be that fast. But they could survive. And I'm seeing Mika, I may have misinterpreted.
Just slightly, basically, the question was how did you do it in Trinidad and Tobago?
Yeah.
Did you treat the water before restoring, or can you do both at the same time? That was basically it.
Yeah, but as I'm saying, it's like it's naturally happening. So it's not that we could really treat the water because it's an annual something. So the corals that are existing in our waters are already conditioned. So the corals that are already surviving the current conditions are already resilient to the conditions.
Thank you so much. On a une dernière question, très courte, parce qu'il faut qu'on arrête. Très très courte, je vous fais confiance.
Bonjour, je suis Marianne Aymar de la Fondation Y-Mer de l'Aquarium de la Guadeloupe. Moi j'avais une question, est-ce que ce sera donc rapide?
I'm working with the Y-Mer. Can you do coral restoration without working on small coral fish that feed on the corals? Because many places did take some measures to protect coral reef fish, but not in Guadeloupe. Can you do restoration without protecting the fish? Thank you.
Yes, because the fish will come back, provided that the reasons for the fish disappearing are taken into consideration. So, for example, if the fish are being overfished, for example, spearfishing or traps or what have you, then of course every effort should be made to reduce those or to have no take zones or reserves. That person should not be overfishing. But the fish will find its way back, especially if you preserve some of the nursery functions of not just the corals, but the seagrasses as well as the mangroves, which is exactly what this project is all about. So you are actually doing it in Guadeloupe through this project. So I'm still very hopeful. I'm positive it will make a difference. May not be, a big shebang of a show, but it's a start in the right direction, and that's to me one of the hardest parts is to get the project going, because it is a long-term investment for something like this.
Thank you, Reia, for all your responses. Et merci mesdames pour avoir participé et animé le débat. Merci
thank you, moderators, and thank you to the public.
Bravo, merci Lou, merci. Thank you, ladies. Thank you so much.